Welcome. That's how I start all my YouTube videos. It's like a little trademark thing I have. So we have a test 2 weeks from today. We meet once next week on Thursday. By the end of that day, Thursday of next week, I want to be done with chapter 18 and done with the material. And then we can basically have kind of a review week with maybe one more little lab activity or at least some review things that will help you get ready for the test. So what I want to do today is look really at chapters 18.1 and point 2, the beginning of it. And to be honest with you, that's going to be most of what I'm going to ask you about on the test. As chapter 18 progresses, it gets really thick with detail and information that I think is just kind of too much for our purposes. It does end, chapter 18 ends with a discussion of kind of a hypothetical model of how exactly cancers arise with more detail than how we've talked about them to this point. So I think on Thursday I'll have a few slides about how cancer forms in humans and relate it to some of the information we talk about today. And then we'll be done with the material for this unit. So chapter 18 is about gene expression. Gene expression. We've talked about the structure of DNA, how it replicates. We've talked about how you go from a gene to a protein with transcription and translation. And now we're talking about how is it that a gene is sometimes showing up and sometimes not showing up, or turned on or turned off. Not every gene in every cell is active. And that's a good thing, because in every single cell in your body, you have all the genes for everything in every cell. Your eyes have your eye cells, the cells that make up your eyes, have genes for liver and kidney function. Your pancreas is made up of cells that carry genes for skin color. But obviously, those genes aren't expressed. Finger development genes aren't expressed in your heart, thankfully, or hopefully not. So there are all kinds of genes in any one cell that is not expressed. So we're going to kind of look at, in general, how this happens. Your book talks about this example of the four-eyed fish, which is a fish that kind of hunts its prey right at the surface of the water, and half of its eyeball sticks out of the water and the other half stays submerged in the water. And the top half and bottom half of the eye of this fish are actually developing in different ways, so that the top half of the eye is good for terrestrial vision or open air vision, and the bottom half of the eye is good for aquatic vision. And how does that happen? How does a fish develop an eye with two kind of drastically different developmental stages? It just depends on which genes it expresses and in which cells. So in the top half of the eye, there are cells that will express only certain genes. And later in the chapter, they talk about transcription factors, chemicals that help get transcription started. If you never transcribe a gene, it never does anything. So basically, transcription only happens in certain genes of the cells in one half of the eye, and then only in certain other genes do you see transcription occur in the cells of other parts of the eye. And that way, the two parts, top half, bottom half of this fish's eye, or fish's eyes, are going to develop in the unique ways that they do. And it's all about gene expression. So, there are two really classic examples of kind of gene regulation and gene expression that you see in an AP biology course. One of them refers to what's called the trip operon, which is what the next slide will be about. And of these 5 slides, the next 2 after this slide are really the 2 I want you to walk away with an understanding of. So this is a general schematic of how E. coli, which is a bacterium that lives in the intestines, that's very well studied, how E. coli controls its production of an important amino acid called tryptophan, which is an amino acid you find a lot or a high concentration of in what? Poultry, that's right. Specifically turkey meat has a lot of tryptophan amino acids in it. Some people think that tryptophan can actually make you sleepy, and maybe that's why people get tired after a Thanksgiving meal. I think it's because they just eat a lot. But this is a schematic of something called feedback inhibition. It's a general look at one example of how cells have mechanisms for controlling how active a gene or genes may or may not be. So to make tryptophan, E. coli bacteria take some starting material, and thanks to a series of enzyme-driven reactions, are able to turn that starting material into this tryptophan that they need. Now these enzymes are produced by cells thanks to certain genes. And this is kind of the first time I've talked to you or shown you anything where we have a gene kind of symbol labeled. You see this a lot, like some sort of like lowercase series with a capital letter. The TRP, what do you think the TRP represents? Yeah, it's referencing the tryptophan that this gene has some association with. In this case, these 2 tryptophan genes, called TRPE and TRPD, work together to make the polypeptide chains needed for this enzyme. This gene is responsible for this enzyme. These genes are responsible for this enzyme. All the enzymes work together to make tryptophan, which is a nutrient that the cell needs. Now, what we found a long time ago in these bacteria was that when the cell has a high concentration of tryptophan, the tryptophan that this process is making feeds back and interrupts both the activity of the enzyme and it tamps down on how activated the genes are. And by interfering with the process that it makes, you can regulate how much tryptophan is produced. And there's a lot of examples of this throughout all sorts of species and how their cells function. So basically, a cell needs tryptophan, it will keep making tryptophan, and then at some concentration where there's enough, that tryptophan will start interfering and block the process that makes it. You use up the tryptophan and its concentration decreases, it stops inhibiting these processes, and more tryptophan can be made. So that's what we call feedback inhibition. Now specifically for tryptophan, we have this diagram that shows how the E. coli bacterium regulates it in a more detailed way. There's a lot going on up here, and I'm going to talk you through this diagram, and then another one, and then show you a fun little picture at the end. So there is there are segments of DNA known as operons. And that word is right here, operon. This is a concept that is kind of above the concept of gene. This is a piece of DNA that contains genes and kind of regulating components as well. Okay? I mean, so far we've just kind of talked about like, oh, a gene makes mRNA, makes a protein, done. Well, an operon involves genes, and here you see these weird gene labels or names. But an operon includes even more than one gene, and then other components that help the cell control how active those genes are. So let's start over here on the left. So a lot of genes are controlled by other genes. In this case, we have what's called a regulatory gene that is going to help regulate the operon. Okay? Usually they're fairly close to each other on a stretch of DNA. Again, this TRPR is the tryptophan gene that helps regulate this whole process. And this gene is going to create a message. What's that process called? When you go from DNA to mRNA? That's transcription. Okay, so we can transcribe this gene to make a segment of mRNA. And then at a ribosome, this mRNA will be read and you will make a protein. Now, this protein is what we call a repressor. It can help stop this whole process that you see behind me. And we're going to talk about how it does that in a minute. So a regulator or regulatory gene is going to make a repressor. In the case of this operon, it's inactive when it's made. It's not doing anything. We'll come back to it. The tryptophan operon, this TRP operon, is going to be made up of some familiar terms. So you've got a promoter. That is where the RNA polymerase is able to bind. What's the special region in a promoter that it really likes to bind to? The TATA box. Very good. That's right, the TATA box. So this is the promoter right here, and it attracts RNA polymerase. The RNA polymerase is going to scan these genes, and as it does, it makes mRNA. Okay, so RNA polymerase is the enzyme that starts transcription, and the result of transcription is mRNA, just like this gene here. Okay? This message is used to make components or entire proteins that are going to help produce tryptophan, which is what we saw in the previous slide. So these are the two parts to one enzyme. This is another enzyme. These are the two parts to a third enzyme. And as we just saw, it's those enzymes that help make tryptophan. So this gene is switched on right now. The gene is on. RNA polymerase is reading the genes and making a transcript, and then making the proteins that serve a function. These proteins are going to make more and more and more tryptophan. Okay? So here's how you keep a cell from making tryptophan indefinitely and maybe even poisoning itself with too much tryptophan. We go back to the repressor, and that's what we look at down here. This repressor that we made is sitting in an inactive state until tryptophan bonds to it. When tryptophan bonds to it, that is what activates the repressor. it literally will change its shape and take on a shape that allows it to bond, what you're looking at right here where it's bonding, bond to what we call an operator. So the operator here is the location where an active repressor can insert itself. When it does that, it's a roadblock. It locks the RNA polymerase down and it can't read the genes. And if it can't read the genes, it doesn't make mRNA, it doesn't make the enzymes that make tryptophan, and the cell stops making tryptophan for the time being. So this is a more detailed look compared to the previous slide. But there are a lot of terms that you want to be familiar with, like repressor, which can, when active, bond to an operator, which can basically turn off transcription by blocking the RNA polymerase, which shows up here at the promoter. Now, what I will probably do is show you either this diagram or the next one, or a whole other operon we never talk about, give you the diagram and have you explain what's happening. And maybe in a written question on the test, a written response question, I could say you must use the following terms, operator, repressor, co-repressor. Tryptophan is called a co-repressor in this scenario because it's what works with the repressor to activate it. Again, the product that all of this operon is trying to make is also the thing that will activate the repressor and stop it, which is a clever, simple mechanic. You want to make sure you don't make too much of something? Well, have a point at which the thing you're making actually goes back and shuts the process down. It's easy for the cell to do something like this. It is not easy to look at this and describe it and to wrap your head around it necessarily, but this is something you'll want to do. There are a lot of questions, there are a lot of AP exams from the past that ask operon questions. And sometimes it's specifically about the trip operon, and this will be in your reading. So let's look at a slightly different operon called the lac. My voice just cracked there. The lac operon. So here, we're going to kind of start, we'll start right here. Here we have another regulatory gene, separate. We have a promoter region where RNA polymerase is attracted to. We have an operator and then genes. But notice, in this case, the operator has a repressor already in place and it's got the gene shut off. So this is a gene that's normally in an off position. Now, it's called the lac operon because the genes that are in this operon code for enzymes that break down a sugar called lactose. So lactose is a sugar. What's a function of sugar in the body? What can you do with sugar?
Turn it into energy.
You break it down and turn it into energy, into ATP. That's right. So there are cells, and again, this is actually found in E. coli. These are very simple cells that use these processes, these specific ones. But E. coli cells come across this sugar. They want to make enzymes to process the sugar. Those enzymes are made, from the transcription and then translation that originates with these genes. So what happens here is kind of the reverse of the previous one. In this case, your regulatory gene makes an active repressor that goes ahead and blocks the process and turns it off. Until some lactose shows up. Now this is, you see this word here, allolactose. It's an isomer, but this basically means lactose. If the cell comes in contact with lactose, some of that lactose can bond to the active repressor and turn it off, which means it no longer fits here, and then what can move forward? The RNA polymerase can now move through and read the genes, make mRNA, which is translated into these enzymes, and you start breaking the sugar down. By the way, what happens when you break all your lactose down and you don't have any anymore?
It stops.
It's going to remove itself. There won't be any lactose to repress. The repressor becomes active again and stops the process. So the first one, the trip operon, is in the on position, and when it's been on too long, the product shuts it down by activating a repressor. And the lac operon, it's off, and when it needs to be on to break down lactose, the lactose inactivates the repressor and switches the operon on. That's a lot more detail than big L is ability to make lactose. Little l is the inability to make lactose or break it down or digest it. So, in our last unit with genetics, you have a gene. This is what the gene does. This chapter is very much about how and when a gene does what it wants and needs to do. Okay. Now, these are the simplest of operons. It can get a lot more complex than this, but in AP biology, we don't deal with that. And there are thousands of different mechanisms that different species and cells have to regulate different things. Chapter 18 goes into it a bit, but we're kind of stopping there. But before we stop, I want to show you one picture to show you how important it is to properly turn on and off the correct genes, especially during development. This is a fruit fly. This is a normal fruit fly that has normal antennae. All the parts of this fly's head and its whole body develop normally because certain genes turn on at the correct time and certain genes are turned off at the correct time and in the correct cells, all based on repressors and other signals, other chemical signals. You can find mutant fruit flies or create them with mutagens like, say, X-rays that have regulatory genes that are damaged and not functioning properly. And in this case, where the antennae should be, this fruit fly has developed legs on its head because of genes that were supposed to repress the leg development genes. They are turned on in these cells, and instead of antennae growing, legs grew out of his face. So this is the kind of, deep importance that gene expression has. Again, your pancreas has the genes for your eye color. It doesn't need to be turning those genes on. Your eyes have the genes to make insulin. They don't need to be making insulin and secreting insulin from your eyeballs. Those things, don't need to be happening, and most of the time, things like that don't happen because of gene expression and regulation of that expression. That's it for our first look at chapter 18. Next class, we will do a wrap-up. You may stop